Meet Louis Vuitton Davari, a 3-year and 11-month-old cockapoo, a mix of a Cocker Spaniel and a Poodle. Today we're showing the early symptoms of Bordetella bronchoseptica, the bacteria that causes kennel cough in dogs, and the treatment of it. Under normal conditions, healthy dogs exhibit consistent feeding behaviors. They usually approach their food bowl with interest, consume meals at a regular pace, and respond positively to treats. Appetite is regulated by a combination of physiological and neurological signals, including ghrelin and leptin, which communicate hunger and satiety to the hypothalamus. One of the earliest clinical signs of Bordetella infection is anorexia, or a reduced appetite. Dogs that usually eat regularly may refuse meals or treats. This happens because the respiratory infection triggers systemic inflation, which causes malaise and lowers their motivation to eat. Similarly, dogs usually spend energy through play and exercise. When infected with Bordetella, however, fever increases fatigue, causing dogs to tire out more easily and rest more than usual. Dogs with Bordetella have a dry, honking cough that often sounds like a goose. This is caused by irritation of the trachea. Now that we've seen the symptoms in the host, we'll look inside the dog's body to see why these symptoms occur. Bordetella bronchoseptica is a gram-negative, rod-shaped bacterium. It attaches to the cilia lining of the dog's trachea, releasing toxins that paralyze cilia, triggering intense coughing. It is an airborne bacterial infection, and dogs can contract it through inhalation of respiratory droplets from infected dogs or contaminated surfaces in shared environments like kennels or dog parks. Once inhaled, the Bordetella bacterium travels through the upper respiratory tract and attaches to the epithelial cells lining the dog's trachea and bronchi, where it sticks, establishing a site of infection and beginning to interfere with normal respiratory function. Specifically, when Bordetella attaches to the epithelial cells lining the trachea and bronchi, the bacteria releases toxins that irritate tissues, damage cilia, and trigger inflammation, reducing the ability of the respiratory tract to clear mucus and pathogens. When Bordetella bacteria enter a dog's respiratory tract, the innate immune system is the first line of defense. When Bordetella bacteria are inhaled by a dog, they first enter the upper respiratory tract, including the nasal passages and throat, before traveling down the trachea and bronchi. The bacteria use specialized adhesion proteins such as fimbriae to attach tightly to the epithelial cells lining the airway, resisting clearance by mucus and ciliary action. Once Bordetella bacteria attach to the epithelial cells of a dog's trachea and bronchi, resident macrophages in the tissue detect them using pattern recognition receptors that bind to pathogen-associated molecular patterns on the bacteria. After recognizing the pathogen, macrophages engulf the bacteria through a process called phagocytosis. While doing this, macrophages also release cytokines, small signaling proteins which trigger localized inflammation, causing blood vessels to dilate and become more permeable, which allows other immune cells as well as plasma cells to migrate from the bloodstream to the infected tissue. Some such cells are called eosinophils, white blood cells produced in the bone marrow which travel through the bloodstream, guided by chemokines, and upon reaching the infected tissue, they become activated. Activated eosinophils release granules containing cytotoxic proteins and kill the bacteria directly. As the Bordetella antigens travel through the tissue, the dendritic cell, the tentacle-like cell, catches and carries small antigen fragments shown here as triangles. The macrophage, blue, also engulfs some bacteria and presents additional fragments. Together, they move through the lymphatic vessels toward the lymph node at the end of the pathway. This lymph node is where the immune system will analyze the antigens, activating helper T cells and B cells, which will coordinate a targeted response against the pathogen. Inside the lymph node, the dendritic cell and the macrophage present antigen fragments using MHC2 molecules, highlighted here in red. These MHC2 antigen complexes bind to helper T cells, allowing the T cells to recognize the pathogen. Once activated, the helper T cell establishes direct cell-to-cell contact with the B cell, signaling it to respond. The B cell differentiates into two types, plasma cells, which will leave the lymph node and secrete antibodies into the bloodstream, and memory B cells, which remain ready for future encounters. At the same time, the helper T cell differentiates into effector T cells, which coordinate the immune response, and memory T cells, which remain in the lymph node or circulate to respond quickly if pathogen returns. This coordinated interaction ensures a fast and targeted adaptive immune response. After activation in the lymph node, plasma cells exit into the bloodstream and begin producing large amounts of antibodies. These antibodies include IgM, the first type released during an immune response, followed by IgG, which provides long-lasting systemic protection in the blood, and IgA, which is transported to mucosal surfaces like the nose, trachea, and bronchi. The Bordetella vaccine was developed in 1988 to protect dogs against Bordetella bronchoseptica, a major cause of kennel cough. By exposing the dog's immune system to weakened or inactivated bacterial antigens, the vaccine helps generate antibodies and memory cells, allowing for a faster and more effective response upon future exposure. The Bordetella vaccination is commonly administered as a subcutaneous injection or a shot, meaning it is given just under the dog's skin. During a secondary immune response, the dog's immune system reacts much faster due to the presence of memory B and T cells formed during the initial exposure. When Bordetella bacteria enter the body again, memory B cells rapidly differentiate into plasma cells, producing large amounts of high-affinity antibodies such as IgG and IgA. These antibodies quickly bind to the bacteria, preventing them from attaching to the airway epithelial cells and neutralizing their toxins. At the same time, memory T cells activate and coordinate the immune response, enhancing macrophage activity and recruiting additional immune cells. Because the response is faster and more efficient, the bacteria are often eliminated before significant symptoms like coughing or fatigue can develop.